Assalamualaikum and a very good day to Dr. Hanis and my fellow friends. We are from Group 1. We'll be presenting about cell chemistry. We actually prepare a mind maps for this topic as it is an effective way to be a better understanding and deliver the LO. So I will start now. The composition of a cell consists of water, which is 70%, and organic molecules and inorganic ions, 30%. So next. The chemical bonds influence the cell biology and also affect the characteristic, for example, structure, size, and function of living cells. The chemical properties of individual cellular components are, for example, the difference between molecules and salt, the polarity of molecules, and also the hydrophilic and hydrophobic molecules. Next, there are two types of bonds. The first one is ionic bonds, which produce salt, and the atom donates or accept electron. And ionic bond also weaker than covalent bonds in aqueous environment. This is because ion also can interact polar water molecules which have positively charged region. Next is covalent bonds. Covalent bonds produce molecules, for example, water, and because covalent bonds is strong, so it needs enzyme to break the bonds. And the covalent bond consists of single and also double bonds. Okay, next. Non-covalent interaction. The chemistry between different molecules in cell involves non-covalent interaction. There are four types altogether. For example, ionic bonds, hydrogen bonds, hydrophobic force, and van der Waals attraction. So as you can see in the figure here, when two molecules interact, more than one non-covalent attraction can take place, and this will increase the strength of the interaction between these two molecules. These two molecules can detach and come into contact again. In easy way, it is on and off process. So next, water dissociation. Water molecule dissociate into hydronium ions and hydroxide ions. As you can see in the figure here, due to the movement of proton from one molecule to another. This process also is a reversible reaction. Next, intracellular pH regulation is maintained at 7 pH by releasing or accepting protons. As you can see in this figure, the example of strong and weak acid and base. A strong acid readily releases a proton, and a strong base readily accepts a proton. So that's all for me. I will pass to the next presenter.
Hi, and a very good morning to everyone. So now I will talk about the small molecule are the building blocks in cell and also the polysaccharide. So basically the left side is the monomer, which is the small organic building blocks of the cell. And the left side will be the polymer, or we also call it a micromolecules, which is the larger organic molecule of the cell that compose of many repeating units of monomer. So by monomer will undergo condensation to form polymer. What is condensation? So condensation is the synthesis of macromolecules by removing the water molecule. Okay, so the polymer also can be broken down to its monomer by undergo hydrolysis, which is the addition of water molecules. So what is organic molecule? Actually, organic molecule is a carbon compound which is contain carbon-hydrogen bond due to the ability of the carbon to form up to 4 covalent bonds readily. So next, I will talk about the polysaccharide. So polysaccharide is the polymer of monosaccharide. And to form polysaccharide, monomers involved are glucose units which are linked by glycosidic bond. So now I will talk about the biological roles. The first one is energy source and storage. Polysaccharide are broken down to monosaccharide to release energy by undergo a series of reactions. Second, cell adhesion. Polysaccharides become glycoconjugated when they becomes covalently bonded to protein or lipids. And glycoproteins and glycolipids in plasma membrane helps to protect cell surface and adhere to one another. They can be used to send signal between and within cells also. So the example of glycoprotein is fibronectin that we have learned in the cell matrix interaction. Okay, second, the cell recognition. So glycolipids on the cell membrane of red blood cell can help us in the recognition of the blood types. So we can recognize the blood type basically based on the combination of sugar. So it is shown by the diagram below. Or the blood type can be determined by the different combination of sugar, 1, 2, 3. So the last biological roles of polysaccharide is protein transport. So the aniline glycosylation is important. By transfer the oligosaccharide side chain that compose of multiple sugar to an SN amino group. So it is usually membrane bound or secreted. So that's all from me. Thank you.
Next, I'm going to explain about the biological roles of the nucleic acid in the cells. First, nucleic acids is the storage of the hereditary information. For example, is the DNA, which store the genetic material. Besides, nucleic acids is also the transient carriers of the molecular instruction. For example, is the DNA, which carries the instruction for the protein synthesis. Besides, nucleic acids is also the source of the chemical energy. ATP provide the energy to drive many processes in the living cells, such as the active transport, reaction coupling, and protein synthesis. In intracellular signaling, GTP or GTP is a source of the chemical energy that will determine if the G protein is the inactive or active. If GTP bound to the G protein, G protein will activated. Whereas if the GTP bound to the G protein is inactivated. It will shown in the diagram below. Next, I'm going to explain more about the lipids. Lipids are amphipathic because it has both hydrophilic and hydrophobic region. It also can be divided into saturated and unsaturated lipids. Saturated lipids are held together by single bonds, whereas the unsaturated lipids are held by double bond. The double bond will create a kinks in order to prevent the fatty acids from packing too close together and keep the liquid in the room temperature, as shown in the diagram below. In biological roles of the lipid act as energy source or storage. For example, is the triglyceride, which are stored in the fat cells and used when energy is needed. Besides, lipid is also important composition in cell membrane. Cell membrane composed of almost 50% of the lipid molecules, included phospholipid, cholesterol, and steroid. In cell signaling, steroid hormone act as a first messenger that initiate the cell signaling by binding to a receptor in the cell. This will cause signal transduction pathway that lead to a cell response. Lastly, in cell recognition, glycolipid on the cell membrane of red blood cell determine the blood type. That's all from me. I will pass my turn to Sufi.
Okay, so I'll be talking about proteins, which are the main building blocks of the cell as they constitute most of the cell's mass. So we go to the biological roles. The first one is that they are involved in cell metabolism, such as hormonal protein, which are insulin that controls the blood sugar level. The next one is protein is also involved in maintaining the cell structure, as fibrous proteins such as microtubules and actin filaments in the cytoplasm help to maintain the shape and stability of the cell. Next, protein also regulates the cell cycle. Cyclin, which is a type of protein, will bound to the cyclin-dependent kinase to perform a cyclin CDK complex so that it can activate and regulate the cell cycle. Other than that, one of the most common roles of protein is that they promote intracellular chemical reaction in the form of enzymes. They also act as signal regulators that relay information from the plasma membrane to the nucleus, such as the G proteins that we learned during the cell signaling pathway. Other than that, protein is also involved in cell motility. They act as motor proteins, such as myosin and actin that are involved in muscle contraction, and also dynein and kinesin that propels organelles through the cytosol. Last of all, protein also help in maintaining the immune system. Antibodies, which are proteins, will destroy pathogens and bacteria in the form of phagocytosis. Okay, next, we go to the protein folding. So as you may know, protein is made up of chains of amino acids that are held together by peptide bond. So this chain must undergo a folding process so that the protein can have a 3D structure, because this 3D structure is very important for the protein to perform its function. So the folding process is influenced by two factors. The first one is the chemical properties of the side chains of amino acid that will determine the non-covalent interaction that is involved. Other than that, the chaperone protein also influences the folding process, as it prevents the formation of aggregates in crowded condition. So chaperone proteins, they do so by exposing the hydrophobic region so that it folds correctly, and it also acts as an isolation chamber that allows folding without any interruption. In either cases, the final 3D shape of protein is still specified by the amino acid sequence. Next is the protein domain, which is the segment of polypeptide chain that folds independently into a compact, stable structure. So each domain consists of 40 to 350 amino acids, and each of them will differ in its function. For example, is the CAP, which has two domains, where one binds to the DNA and the other binds to the cyclic AMP. So the binding to these two domains will promote the expression of genes. Other than that, the biological properties of protein molecule depends on its physical interaction with other protein, meaning that protein must bind to other molecules to perform its function. That's why every protein has a specific ligand binding site. However, this ligand binding site can be destroyed if there is an incorrect folding or changes in the amino acid sequence. Other than that, the binding of protein to other molecules shows great specificity, where they can just bind to a few or more molecules. This specificity can affect the strength of interaction. So tight binding occurs when the ligand has matching surface with the protein. Therefore, it will form many non-covalent bonds which are strong. So the binding of these two will be in a long period of time. Other than that, conversely, the weak binding will occur due to the poorly matching surface between the two molecules. Therefore, only a few non-covalent bonds are formed, and therefore they normally dissociate rapidly due to the thermal motion. So to conclude all, the cell chemistry plays a vital role in cell biology, whether in terms of its composition and interaction. Cells are made up of four main subunits, which are the sugar, fatty acid, amino acid, and also nucleotides. These subunit will undergo condensation to reduce macromolecules that are involved in many cellular processes in the body. So that is it. Thank you.